Hi, everyone, and welcome to Principles of Microbiology. This is the first lecture in this course, and we will be covering Chapter 1, Introduction to Microbiology. Hopefully, at this point, you've all had a chance to set up Connect access. That simply can be done by just clicking the MH Connect button in Canvas and following the directions. If you don't have first day access, it'll allow you the prompt to purchase Connect directly through that McGraw-Hill or MH Connect button. Anyway, let's get started. So microbiology um, is an area of biology. It is a subset of biology, and it's going to deal with organisms that are too small to be seen with the naked eye, as the name implies, microbiology. And you'll actually need to use a microscope to see most of these organisms. Microorganisms or microbes are often referred to as germs, viruses, agents, or even just simply as bugs. Only a small minority will actually cause harm to us as humans. Most microbes that are out on this world actually have nothing to do with you. And there are quite a few that are beneficial to you. But there are also ones that are very bad for you and can cause lots of issues for you. And the purpose of this course is to kind of really dive into not just the bad ones, which I'm sure we're all familiar with some, and there might be others that you're not familiar with, and also how to prevent those microbes from causing infections. What are the preventative measures that we have? And then also treatment options if you should become infected. Not all these infections can be treated. A lot of times it is dependent on your immune system sort of kicking in and doing its job. But we do have antibiotics, antifungals, and antivirals that are used to treat various types of microbial infections. One other aspect that we will consider throughout this course is drug resistance. As when we talk about treatments, some of these drugs can develop resistance, which can be problematic in the clinical setting for treating some of these infections. But that's the bad guys. There are also plenty of good guys. For the past 2 decades, almost the past 2 decades, there has been what we refer to as the microbiome project. And for the last decade, this has all sort of been centralized. There has been a lot of beneficial information that has come out about microbes, especially ones that have positive effects on our human body. And we'll dive into some of those as well throughout the course. And then we will talk about the ones that also do not have anything to do with you, um, which is most microbes. They can infect other types of animal species. They can infect other types of living organisms on this planet. One thing that you will come to find out is that a lot of microbes are very specific for the host that they tend to infect. So we have various types of cellular microorganisms. This will include bacteria and archaea. These are both going to be prokaryote cells. We then have fungi, protozoa, and helminths. These are all going to be eukaryotic-based cells. The helminths really are not technically microorganisms because these are your worms, and they can be several feet long, and these are things that you can actually see without the aid of a microscope. But there's really nowhere else to shove them. So we kind of just group them in under microorganisms. And then we also have our non-cellular microorganisms. These are non-living entities, and this will include your viruses and your prions. Now, microbes are oftentimes easy and can also be difficult to study. They reproduce rapidly. Large populations can be grown in a laboratory. If you're talking about a bacteria like E. coli, you can go from one cell to hundreds of billions of cells within an 18-hour period. So if you're looking for mutations, if you're looking to study drug sensitivity. This is where it can be very easy because you can end up with populations of cells very quickly and look for changes very rapidly. It can be difficult depending on the level of biosafety that's required for the cells. Um, if you're talking about the non-cellular microbes, such as viruses, they will need to be transfected into a host cell in order to really truly study them. So that's where it can get a little more difficult because not only are you looking at the virus itself, but now you're looking at, excuse me, now you're looking at, growing mammalian cells or human cells, cell lines in order to be able to study these things. Microbes cannot be seen directly. They must be analyzed through indirect methods, such as using a microscope. Any of your prokaryotic and eukaryotic cells will most likely be able to be used with this or viewed with a standard light microscope. However, when you start getting into more detailed structures, if you start getting into viruses and prions, you will need to use an electron microscope or something a little more powerful. Now, microbiologists will study cell structure and function, really just mainly for drug discovery, and also for host pathogen interactions, meaning or pathogenesis, how can this organism cause disease? We'll look at the growth and physiology, the genetics, taxonomy, and its evolutionary history, as well as interactions with living and non-living environments. All of this will help with not only drug discovery, but that whole process of pathogenesis. There are several branches of microbiology. That, believe it or not, there is more to this field outside of the clinical setting. We have medical microbiology, which is why most of you are here. This deals with microbes that cause disease in humans and our animal counterparts. We also have public health microbiology and epidemiology. This job of these particular types of scientists is to monitor and control the spread of disease in communities. This would include the U.S. Public Health Service, the Centers for Disease Control, which is actually one of the most preeminent organizations for microbial and disease study in the world. And it is a member of the World Health Organization, which is really just sort of the larger umbrella that the CDC sort of falls under. Immunology. This is the complex web of protective substances and cells that are produced in response to an infection. This field will also include the development and administration and monitoring of vaccinations, blood testing, and allergies. We also have industrial microbiology. This is the safeguards for our food and water. Biotechnology, as well as microbes used to create amino acids, beer, drugs, enzymes, and vitamins. For example, acetic acid, which is vinegar, is produced by microbes. Fermentative processes for our water safeguards. A lot of purification of water systems use microbes to actually break down organic waste within the water. So this is actually a very large field within itself. Agricultural microbiology, this is the relationship between microbes and farm animals, as well as crops. Plant specialists and animal specialists will get involved here. Up until the early 2000s, a lot of antifungal drugs that were used to treat crops were the same family of drugs that were used clinically. That started to pose to be a little bit of a problem with drug resistance because we had low levels of exposure to the drugs by fruits and vegetables that we were eating, which allowed a lot of these fungal species to develop low level resistance, which allowed it a nice stepping stone into full-blown resistance. That has since been rectified. There are different classes and families of drugs that are now used for ag versus clinical. They weren't the same drugs before, they just belonged to the same families, which gave for a lot of cross resistance. Environmental microbiology, the effects of microbes on the Earth's diverse habitats. How do shifting microbes or patterns of microbes affect the overall habitat? Aquatic soil and geomicrobiology and astrobiology are all subfields under this. So the overall impact of microbes on Earth is microbes have shaped the development of the Earth's habitats and evolution of other life forms for billions of years. Soon after the Earth was formed, the first ancient cells formed, and what we slowly start to see where bacteria and archaea develop. About a billion years after the first cells started to appear, those bacterial cells, we started to see some eukaryotic cells start to pop up. And that brings us to the eukaryotic cell, which is something that you guys probably have studied extensively, if not in your prior education, but here with taking Gen Bio with uh, Gen Bio courses. Essentially, cells with a nucleus are classified as eukaryotes, and the prefix eukaryote means true nucleus. Cells without a nucleus are classified as akaryotes, and bacteria and archaea have no true nucleus. So here you can see just a little timeline going back about 5 million years on when things started to sort of pop up from the origins of the Earth, all the way up to present time, which is where we kind of fall into this timeline here. So let's start off with bacteria. First of all, bacteria are ubiquitous. Bacteria are found everywhere. Okay. Any surface, they're found. They are found deep in the Earth's crust. They're found in the polar ice caps, so you can even find bacteria in very extreme environments. The oceans, as well as inside the bodies of plants and animals. On surfaces, I mean, you guys are well aware of this, that maybe you're not, that microbes can be found in the air, they can be found on surfaces. And oftentimes, if you have a cut or a scrape, you need to clean that out well. Mainly because A, you have a break in a barrier, but also that there's microbes on the surface of your skin. There's microbes maybe on the surface that pierce the skin. So those are all things that need to be taken into consideration. Um, as far as your own body is concerned, um, the human body is inhabited with lots of microbes or microbiome. And they are going to inhabit most of your surfaces. This would include your skin. And believe it or not, your skin is not actually the most hospitable place to live for a microbe because of the acid mantle and the high salt concentration. It actually retards the growth of a lot of things. But you also have mucosal membranes, which would include oral, nasal passageways, oral passageways. So the whole ear, nose, throat region, your um, entire GI tract from the oral cavity all the way to the anal rectal area. Uh, Respiratory tract, especially the upper respiratory tract, you'll find lots of microbes. And then in females, the vaginal tract. And these are all surfaces within the human body. And just one little fun fact, it is estimated that you have over 100 trillion bacteria in your large intestines alone. That's a very large number. It's even more impressive considering that you are made up of only about 10 trillion human cells. So you actually have 10 times more bacteria in your large intestines alone than you actually have of human cells. Now, evolution, this is a concept that hopefully you have been taught somewhere along the line. This is the accumulation of changes that occur in organisms as they adapt to their environments. The theory of evolution is documented in every day in all corners of the planet. And it's an observable phenomenon that's testable by science. And it's something that can be easily observable with microbes, given that they have very fast population turnovers as far as time is considered. Microbes are deeply involved in the flow of energy and food throughout the Earth's ecosystems. Bacteria invented photosynthesis and oxygenic photosynthesis, and the later evolution of oxygenic photosynthesis. The production of oxygen by microbes allowed species to diversify to the point where we have aerobic organisms undergoing processes of cellular respiration. Photosynthetic microorganisms contribute the majority of the oxygen in the atmosphere, and this is accomplished by bacteria and algae, which account for more than 70% of the Earth's photosynthesis. Microbes also play a very important role in decomposition. These are going to be your heterotrophs, where they break down organic material. This would be the breakdown of dead matter and waste products, and it's accomplished by bacteria and fungi. Microbes are all the main forces that drive the structure and content of soil, water, and atmosphere. Uh, you get gas production by microbes, microbes that are living within the Earth's crust, and bacteria and fungi living in complex associations with plants and animals. Humans have been using microorganisms for thousands of years to improve life and even shape civilization. For example, bread, wine, beer, things most of us have enjoyed throughout our life, is produced by yeast. Other fungi are used for cheese production and even some bacteria. Moldy bread was used in Egypt to treat wounds. We now know that penicillin was derived from mold. Um, Biotechnology, this is manipulation of microorganisms to make products in an industrial setting. Recombinant DNA technology, this manipulates the genetics of the microbes, plants, and animals to create new products of genetically modified organisms. For example, human insulin is something that is made using yeast cells these days. Yeast are eukaryotic cells, so they can end up pumping out lots of this protein. And what they've done is they've genetically manipulated the organism to have the human insulin gene in order to make insulin. And this is how they could do this, along with a lot of the derivatives or second, third, fourth generation insulin products that are out there. Bioremediation, this is the introduction of microbes into the environment to restore stability or to clean up toxic pollutants. Shortly after the BP spill, there was a huge study that showed, and the BP spill may have occurred well before many of you were born. It was an oil spill in the Gulf of Mexico. But what they found was, BP tried to clean that up, but most of it actually dissipated. And it actually was broken down by microbes within the Gulf, the water. And that is something that they now use. Bioremediation is something that's used to break down organic material in drinking water. A lot of water treatment plants will have these. A lot of your larger filtration systems will also have what they refer to as biomasses that the water will pass through and organic material will get broken down. A lot of organic waste is used or broken down um, by using microbes. So it's a way to really sort of remediate a lot of these organic materials. We also then come to pathogens. Pathogen is any agent, such as a virus or bacterium, fungus, protozoan, worm, helminth, that causes diseases. There are nearly 2,000 different microbes that can cause disease. And what with this is with pathogens is they will cause tissue damage for you. Okay. There's another term referred to as opportunistic pathogens. With opportunistic pathogens, the default is not to actually cause disease, but rather to live in a symbiotic relationship with a host. However, if environmental conditions should change, they can become pathogenic and cause disease. Let me give you a few examples. One is we have normal microbiota throughout the human body. When you have a diabetic patient who is not actively regulating their blood sugars, um, that leaves for more sugar within the interstitial fluids and within the blood. And that can lead to an increase in infections for diabetic patients. Another example is if you have a female that goes on an antibiotic, broad spectrum antibiotic for some infection, let's just say, it's an upper respiratory infection, goes on a broad spectrum antibiotic. Unfortunately, that antibiotic doesn't discriminate against the bad guys within the lungs causing the upper respiratory infection, but rather it just kills broad spectrumly. That's why it's called broad spectrum antibiotic. So it kills not only the bad guys, but also kills the good guys. And what can oftentimes arise is a yeast infection within the vaginal tract. And what's happening there is because of the antibiotic, a lot of the good bacteria within the vaginal tract has died off, which means there's more available resources or food, as well as the dead bacteria now, which the yeast will take advantage of. They will have more nutrients, they will grow more, and now your numbers of yeast are not in what we call acceptable ranges or normal biota ranges, but now in infectious ranges. So that's an opportunistic pathogen. Usually there's some type of environmental change that leads to that particular type of organism causing disease. If it's just straight up pathogen, it will cause disease. If we look at the US top causes of death in all diseases, you can see that COVID-19 was one of the major ones on the microbial side of things, but the rest of these, heart disease, cancer, accidents, chronic respiratory diseases, that's a big one for microbial, Alzheimer's disease, diabetes, kidney disease. A lot of these actually are not microbial related. So we have COVID-19, we have the lower respiratory infections as well as flu and pneumonia. If you look worldwide, that actually shifts. We see a lot more microbial diseases in worldwide causes of death than we do in the US. And the big reason for that is A, we have robust vaccination programs in the US for now. We also have very high standards for food. We have clean water. We have access to clean water. And all that plays a huge role, which you do not see in some very highly populated developing countries, where a lot of these infections can really sort of, come through, such as diarrheal diseases, flu, pneumonia, um, being some of the main ones here. Infectious diseases, these are new and emerging diseases, as well as older or re-emerging diseases are increasing. We've seen SARS, Ebola, AIDS, hepatitis C, virus and viral encephalitis, COVID. More recently, flu is something that we see every year. These are all things that sort of are infectious, and they sort of either emerge or re-emerge. For the most part, measles, polio, leprosy, parasitic worm diseases have largely been eradicated. And again, I put in there for now. They've been mostly eradicated because of very robust vaccination programs. And unfortunately, there are some cracks in our vaccination programs that we've been seeing recently. We've seen measles sort of spike up. We're starting to see some other things sort of pop up, like whooping cough, which these are things that we should not see again. And unfortunately, it's due to a lot of misinformation around vaccines. Hopefully, some of which I can clear up for you guys throughout this course. Diseases that were once considered non-infections. Certain diseases once considered non-infectious are now found to be caused by microbes. For example, gastric ulcers. We now know it's not just something that happens. It's actually caused by a bacteria called H. pylori. And one thing that can sort of really push H. pylori to causing these ulcerations is stress. The link between certain cancers and bacteria and viruses, especially viruses, hepatitis viruses are a big player there. Also, HPVs are human papillomaviruses with certain types of cancers. Sorry, Coxsackie virus has been associated with diabetes and schizophrenia. MS, OCD, coronary artery disease, obesity. These can also be linked to chronic microbial infections. We also know that a lot of microbes aren't as direct with their infections or side effects. And today, we are discovering the subtler side of microorganisms and the quiet, slow, destructive diseases that they can cause. For example, female infertility can be caused by chlamydia infections. Chlamydia infections that might go unnoticed for years, if not decades, slowly causing damage to a lot of the internal structures involved in fertility. Liver cancer can be caused by hepatitis viruses, especially with several hepatitis flare-ups. Cervical cancer, not only cervical cancer, but penile, anal, throat, prostate cancers as well, caused by HPVs. We also have an increasing number of patients with weakened immune systems that are subject to infections by common microbes not pathogenic to overall healthy individuals. And the reason for this is actually a lot of advancements in medicine, especially with cancer treatments. Unfortunately, immunotherapy and immune suppression is something that is being used more frequently. And that does put people at risk of developing certain types of infections. MS treatments are immunosuppressive. You also have transplants have been increasing significantly, which involves the use of immunosuppressive drugs. So we're seeing a lot more immune suppressed patients. Drug-resistant microbes are also contributing to the increase in infectious diseases. So there are three basic cell lines that appear during the evolutionary history, differing in complexity, contents, and function. These are the archaea, eukarya, and bacteria. Majority of microorganisms are single-celled. All bacteria and archaea are going to be single-celled, period. Some eukaryotes are single-celled. And then you also have some eukaryotes which can form helminths, which are the worms. And then some fungi can be multicellular as well. For example, a lot of the molds are multicellular and are not single-celled organisms. Bacteria and archaeal cells are about 10 times smaller than eukaryotic cells. They do lack organelles, membrane-bound organelles. They can contain small double membrane structures that perform specific functions. That's what organelles are, in case you uh, didn't have that prior. Prokaryotes will not contain these structures, but eukaryotes will. For example, ER, lysosome, peroxisome, um, um, Golgi, Um, these are all going to be examples of those membrane-bound organelles. The eukaryotic cells will contain these organelles. They will also contain a nucleus. They may have a mitochondria or a chloroplast. We'll get into a little bit more when we hit chapter 4 about the history of mitochondria and chloroplast, but that's a little bit of a spoiler right now. They were once prokaryotic cells that were obtained by early eukaryotes through a process called what's referred to as the endosymbiotic theory, which essentially states these early eukaryotes essentially gobbled up these prokaryotic cells for energy purposes. Um, Acellular microorganisms, these can be your viruses. This is composed of a small amount of genetic material. It's either DNA or RNA. You'll never have both. Wrapped up in a protein covering coat, and about half of viruses will also have a lipid membrane. That lipid membrane will actually come from the host cell. Prions, these are small proteins folded in intricate ways, which sometimes behave like microorganisms, and they are transmitted from one human to another, we think. That still has an asterisk next to us because we're still trying to figure out exactly what prions are, their overall impact, etc. Here's some examples of microbes. We can see a helminth, then we can see a fungi, protozoan, bacteria, virus. What you're looking at there is herpes simplex, and even a prion. We don't have any archaea here. We're really not going to talk too much about them because A, they really have nothing clinically relevance, and they're really kind of just single-celled boring organisms. We really don't get too much into them in this course in particular. So overall, going back over some history here, some early ideas about disease transmission. Certain food spoiled, became inedible. This oftentimes can cause illness. The black pig, plague, and smallpox are caused by some kind of transmissible matter. There was also a belief in spontaneous generation. These were all early ideas about disease transmission. As far as the spontaneous generation, Pasteur kind of put this to bed with his experiments. He essentially set up a broth that had been boiled, creating a sterile-free broth. One of them was exposed to air, one was not. The one that was developed organismal or developed growth within the meat broth. The one that was not exposed to air did not. Saying that, you know, essentially they were obtained from somewhere and not that they just spontaneously developed out of the broth. Robert Hooke was one of the first guys to observe microbes in the 1600s. That was followed up by Leeuwenhoek, who made a crude microscope, which really allowed us to sort of springboard into seeing a whole new world of things. Your more modern microscopes are more refined lenses, a condenser, finer focusing devices, as well as built-in light sources or other energy sources that are used. Typically, student microscopes are not greatly different in structure and function than the early microscopes. The basic standard light microscope is what it was 400 years ago. But, you know, it works. Light does have its limitations. As far as microscopy is concerned, we'll cover more about this in chapter 3, but essentially you are kind of capped at 2000x for light microscopy. And that's really due to a resolution issue to be able to distinguish between 2 points because of the limitations of light. I'm not going to dive too much into that because physics is not one of my strong points, but if you've had a physics course, you might be able to sort of dive into that a little more deeply. In the 1980s, we had the invention of the PCR technique. In the 2000s, the importance of small RNAs. And this really opened up the whole identification, genetic identification, using both PCR, small RNAs. For that, a lot of Identification today is either done by um, antigen antibody interactions, such as ELISA assays, or QRT-PCR analysis of genetic material to get quick answers on what the organism is. 2013 opened the door to CRISPR technology, and it's pretty amazing. When I was in grad school, sequencing a genome took months. Now sequencing a genome maybe takes your lunch hour. And you can get whole genome sequencing done pretty quickly. So it's quite amazing to see a lot of what's been done on the tech side. Early experiments showed that microbes are everywhere. Air and dust are full of them. The entire surface of the earth and its waters and all objects are inhabited by them. Some of you in your bio courses may have done what they call microbes are everywhere kind of lab, where you kind of take a swab and you swab something surface and put on a petri dish to see what grows up. And something should grow up. We may do this lab. I typically will have students swab their cell phones, as that tends to be the dirtiest thing that now people carry on their person. It used to be money, but cell phones have sort of taken that number one slot. Um, the discovery of spores and sterilization. Um, John Tyndall found that microbes in dust and air have a high heat resistance, and this really became the discovery of spores. Later, the discovery and detailed description of heat-resistant bacterial endospores clarified why they were resistant to heat. Um, and um, they can be a little more complicating as far as, prevention. You do have to be a little careful sometimes with spores. You have to make sure you're taking full precautions, or full sterilization techniques are in play. Um, in order to be considered sterile, you are completely free of all life forms, including spores and virus particles. And one thing that I want to stress with this is that you are completely free of all living life forms and active viral particles. That's not to say that you might not have a, dead bacteria that's still part of the mix in a sterile solution. In fact, a lot of media that's used for micro will have dead yeast as part of an ingredient. Um, Obviously, the yeast cells are there. Uh, the dead yeast cells are used for nutrients, but they're not alive, so they're considered sterile. Robert Koch, he linked the specific microorganisms as far as causing specific diseases. And we'll actually talk a little bit later when we get to pathogenesis about Koch's postulates. Dr. Oliver Wendell Holmes observed that mothers who gave birth at home experienced fewer infections than mothers who gave birth in a hospital. And again, more exposure to microbes in a hospital setting. Joseph Lister, he was the first to utilize hand washing and misting operating rooms with antiseptic chemicals. The techniques became the foundation for modern microbial control, and a lot of these are still used today. And actually, his namesake is found on a very common product, Listerine, which is a disinfectant. Pasteur invented pasteurization, along with many of his other impressive contributions. And he also showed that human disease could arise from infection. And again, Robert Koch established a series of proof that verified the germ theory of disease, meaning that specific microbes can cause disease. Naming, classifying, and identifying microorganisms. Nomenclature. The overall assignment of scientific names to The various taxonic categories and individual organisms is referred to as a nomenclature. Organisms are classified in attempts at the orderly arrangement of organisms into a hierarchy. And identification is the process of discovering and recording the traits of organisms so that they can be recognized or named and placed in a taxonic scheme. As far as naming is concerned, we have what we refer to as binomial system. And this is going to include two names in the whole naming scheme, and that's going to be the genus and species. The genus name can be abbreviated to save space if the genus name has already been stated. So for example, instead of saying Staphylococcus aureus, it can be abbreviated as Staph aureus. Okay, and that is the end of chapter 1.